Thank you for joining me on this adventure. Your presence means the world to me. Hello and a warm welcome to our channel. Today, we're delving into Ferroplasma. Ferroplasma is a genus of Archaea that belong to the family Ferroplasmaceae. Members of the Ferroplasma are typically acidophillic, pleomorphic, irregularly shaped cocci. The Archaean family Ferroplasmaceae was first described in the early eras. To date, very few species of Ferroplasma have been isolated and characterized. Isolated species include Ferroplasma acidiphilum, Ferroplasma acidarmanus, and Ferroplasma thermophilum. A fourth isolate, Ferroplasma cupricumulans, was later determined to belong to a separate genus. All known Ferroplasmaceae are iron-oxidizers. As we move forward, let's uncover the untold stories and fascinating intricacies of cell characteristics and physiology. Ferroplasma cells are pleomorphic and lack a cell wall. All known members of the genera are acidophils that thrive in environments with ranges from 0 to 2. They are also mesophilic to moderately thermophilic with optimal temperatures ranging from 35–55 C. Tetraether-based lipids are an important part of the Ferroplasma cellular membrane and our cells to maintain a gradient. A study of Fahrenheit acidarmanus found that cytoplasmic was maintained while the environmental ranged from 1.2. Variations in the tetraether lipids of the family Ferroplasmaceae are used for chemotaxonomic identification at the genus and species level because many members possess identical RNA sequences. Members of the genus Ferroplasma are chemotaxotrophs that can oxidize ferrous iron to acquire energy, but despite evidence of carbon fixation, lab cultures often require an organic carbon source such as yeast extract for growth. In the absence of iron, some lab-grown strains have been capable of chemogonotrophic growth. The time has come to unravel the secrets behind ecological importance and gain a deeper understanding. Iron is the fourth most abundant mineral in Earth's crust. As iron oxidizers, Ferroplasmaceae participate in the biochemical of iron. Ferroplasmaceae are often identified at acid mine drainage (AMD) sites. When ferrous iron Fe is oxidized to ferric iron Fe at mine sites, Fe spontaneously reacts with water and iron-sulfur compounds like pyrite to produce sulfate and hydrogen ions. During this reaction, ferrous iron, which can be utilized by Ferroplasma, is also regenerated, leading to a propagation cycle where Fe is lowered. The reaction can be described by the following equation: Fis plus a Fe plus Tho a Fe plus So plus Sh. Ferroplasma species are often present at AMD sites where they participate in this cycle through the biotic oxidation of ferrous iron. Ferroplasmaceae may have important applications for bioleaching metals. Microbial bioleaching occurs naturally in the highly acidic environments that are home to Ferroplasmaceae. Harnessing the power of bioleaching to recover metal from low-quality ores and waste material is energetically advantageous compared to smelting and purifying. It also produces fewer toxic byproducts. Studies have shown that the inclusion of Ferroplasma thermophilum along with the bacteria Acidophilus caldus and Leptospirillum ferrophyllum can augment the leaching process of chalcopyrite and increase the rate at which copper is recovered. Let's now shift gears and explore Ferroplasma acidiphilum through a critical lens, uncovering its strengths and weaknesses. Ferroplasma acidiphilum has been shown to grow as a chemotaxotroph and to grow synergistically with the acidophilic bacteria Leptospirillum ferrophyllum. The strain Ferroplasma acidiphilum yet is a facultative anaerobe with all the required genes for arginine fermentation. Although it is unclear whether Ferroplasma acidiphilum yet uses its arginine fermentation pathway, the pathway itself is an ancient metabolism that traces back to the last universal common ancestor LUCA of the three domains of life. Get ready to immerse yourself in the world of Ferroplasma acidarmanus as we examine its impact and relevance. Ferroplasma acidarmanus was isolated from mine samples collected at Iron Mountain, California. Iron Mountain CA is a former mine that is known for its acid mine drainage (AMD) and heavy metal contamination. In addition to being acidophilic, Fahrenheit acidarmanus is highly resistant to both copper and arsenic. Let's now enter the realm of Ferroplasma cupricumulans, formerly Ferroplasmaceae, and discover the fascinating stories it has to tell. Ferroplasma cupricumulans, formerly Ferroplasmaceae. In 2006, Ferroplasma cupricumulans was isolated from leachate solution collected from the Myanmar Ivanhoe Copper Company MICCL mining site in Myanmar. It was noted to be the first slightly thermophilic member of the genus Ferroplasma. However, in 2009 a new genus of acidophillic, thermophilic Archaea, acidophilum, was identified. It was proposed that, based on the similarity and DNA hybridization, be transferred to the genus Acidiphilum and renamed Acidiphilum cupricumulans. As we progress through this video, let's shift our attention towards Ferroplasma thermophilum and uncover its hidden depths. In 2008, Zhu et al. Described the isolation of the organism Ferroplasma thermophilum learnt from a chalcopyrite column reactor that was inoculated with acid mine drainage AMD from the Day copper mine in China's Hubei province. In aerobic conditions with low concentrations of yeast extract Fahrenheit, thermophilum grows by oxidizing ferrous iron. However, in anaerobic conditions Fahrenheit, thermophilum reduces ferric iron and sulfate. This makes Fahrenheit thermophilum ecologically important for iron and sulfur cycling at pyrite-rich mine sites. I love hearing from you, so leave your thoughts and suggestions in the comments.